Excellent. Thank you, David. Excellent. So yeah, excited to talk about our R-loop work today. So my lab really focuses on how RNA binding proteins impact development and how this is important in a disease context. And so we study splicing for splicing's sake, and then on the other side, we study how splicing aberrations and aberrations in other RNA binding proteins might dysregulate R-loop structures, which are these, which is what I'm going to tell you about today, really focusing on this R-loop stuff. So what is an R-loop for those of you who don't think about this all the time? So what R-loops are is they are endogenous structural variants that form in DNA. And so this is what an R-loop looks like. So it is made up of two components, an RNA-DNA hybrid component, as well as a single-stranded DNA. It is thought the most prevalent model of how these form is that they form through when the nascent RNA exits the RNA polymerase, they thread back onto the exposed DNA, and this forms an RNA-DNA hybrid. And because this structure is formed, the single-stranded DNA cannot rehybridize to that other DNA strand. This is not the same as the RNA-DNA interaction that occurs inside of the RNA polymerase catalytic subunit, right? So by de facto, inside of the RNA polymerase, there's RNA forming that is base pairing to the DNA as the RNA is being produced. That is not what an R-loop is. That is protected inside of the core of the RNA polymerase. What an RNA-DNA hybrid that's part of an R-loop structure is something that happens outside of that unit, okay? Just so that you distinguish, it's not every transcriptional unit that forms an R-loop, just sometimes they happen, sometimes they don't, and the context in which they form is still up for debate, and so we can happy to talk about that at length if anyone is interested. But what do we know about R-loops and why should you care? So these are things that happen, they're non-structural variants. And the reason that we got interested in it is because R-loops have been implicated, although really what they're doing in these diseases is not that clear. So R-loops have been implicated in several neurological diseases, and so there are R-loop regulating factors that are mutated in ALS, also in a different ataxia form called AOA2, ataxia ocular, I'm going to butcher the name, like he said, hematopoiesis. We'll just say AOA2. Also, Fragile X syndrome. So there's several neurological diseases where R-loop regulating factors or R-loop promoting sequences are linked, but what exactly those R-loops are doing in that context is still not completely clear. We came at, my background is primarily hematopoiesis, and so we came at it from a hematopoietic perspective. So there's been some recent work implicating this in a hematologic malignancy called myelodysplastic syndrome. There's also forms of autoimmune diseases like Acuti-Gutierrez syndrome, which also have R-loop regulating factors that are mutated. But again, why does this mutation, this R-loop regulating factor, lead to this phenotype in humans is unclear. It's more of a correlation at this point. And then recently, there's been evidence linking R-loop dysfunction to various forms of cancer. But like I said, what's really unclear under all of these contexts is that there's these correlations but no causation. So what I hope I can convince you at the end of this talk is that what we've tried to address is what aspects of R-loop dysfunction are contributing to diseases and how can we use an organismal context to address this question. So why would this be a hard question to answer? And that's because R-loops have really pleiotropic functions in the cell. So they're not just byproducts of transcription that form, they actually are thought to have functionality and gene regulation. So for example, it's been shown that R-loops can form and regulate epigenetic modulation. R-loops in promoter proximal regions can inhibit DNA methyltransferases and therefore change the prevent methylation at a promoter and keep a gene on. It's also been shown that if you have excess R-loops, again, that could be bigger R-loops that already exist or R-loops that form in new places, this can lead to RNA-DNA hybrids are more stable than DNA duplexes, and so this could be deleterious during DNA replication, where the replication machinery gets stuck and it kind of hits this R-loop and can't move forward. This will lead to DNA double-strand breaks and genomic instability. Other groups have shown that R-loops are important for DNA repair and maintenance, and so again, how they're functioning in each context is going to be dependent upon the chromatin context in which they form. Within the cell, there are a host of machinery. So what I hope I can convey also at the end of this is that the balance of R-loops, so too many is bad, too little is bad. If you have too few, you may have genes aberrantly getting methylated and get turned off. If you have too many, you're going to end up with genomic instability. And so there's several flavors of proteins that are important for maintaining the balance of R-loops. So you'll have helicases that are important for unwinding RNA-DNA hybrids to remove them from the cell. There's a point of no return type of mechanism. RNase H enzymes will cleave and destroy the RNA moiety in the RNA-DNA hybrid, and so you eradicate the R-loops that way. Topoisomerases are also thought to be important as they unwind and change the torsional stress within the chromatin so that R-loops aren't prone to form. So they are prone to form in certain torsional stress context. And as well as splicing factors. And the idea with splicing factors is that if you see here, for example, kind of the first factor identified to regulate R-loops was SRSF1, and this was our first splicing factor. If you remove splicing factors, the idea, still an idea, not proven, but the idea is that you now have more naked RNA, which now can loop back in more, it kind of changes the dynamics of in which it can loop back in and hybridize to the DNA. But really, how splicing factors regulate R-loops is still up for debate. So basically, I'm going to talk about two stories of how we looked at how splicing factor regulated R-loops might impact neuronal function and development and survival. And the second part is looking at a RNA-DNA helicase that's important in the hematopoietic system for regulating how hematopoietic stem cells emerge. So we use the Zebrafish as our model system to study R-loop dynamics in an in vivo context. And at first, you may think a zebrafish is not the same as a human being, but I hope you'll believe me and trust many others, is that, the cell types and the ways that cell types form and the ways these tissues develop in a zebrafish are highly analogous to what happens in a human context. So the neurons are highly similar, blood cells are highly similar, and especially during development, these processes are highly conserved. All right, so I mentioned basically the talk is split up into two parts. Feel free to ask questions during or after, because it's going to cover a few different things. So the first part is going to focus on a splicing factor called Sf3b1, which is part of the U2 SNRP complex. And all of this is going to be centered on looking at how R-loops impact neuronal development and survival. The second half of the talk, I mentioned I'm going to talk about this helicase called DDX41 and how this helicase how the R-loops that are regulated by this helicase will impact hematopoiesis. So two organ systems and two different factors. So definitely ask questions if I lose you. So what is Sf3b1? So Sf3b1 is a crucial splicing factor. As I mentioned, it's part of the U2 SNRP, which is important for binding the branch point site. It's an essential gene in all cells. It's important for splicing, not just intron removal, but all of splicing. What Sf3b1 is best known for, as I mentioned, it binds to the branch point site and helps to define 3' splice site recognition. And Sf3b1, the reason that we got interested in it is because this was found to be mutated in myelodysplastic syndrome in about 2011, 2012, but no one really understood why would mutations in this splicing factor lead to hematologic malignancies. So these are at least three different, so just like R-loops are pleiotropic, so is Sf3b1. So I just mentioned the most canonical, well-understood functionality of Sf3b1 in splice site recognition. There's also some work implicating Sf3b1 and other splicing factors in regulating transcriptional elongation. And then work by many people, including our work that I'm going to tell you about today, have shown that Sf3b1 may be important for regulating R-loop levels, which is important for maintaining genomic integrity. A little bit blown out, sorry. So what we're looking at here, so to mention that we use zebrafish, and so we're looking at here are zebrafish mutants for the factor for Sf3b1. On your left here is the picture of a normal wild-type sibling animal that is about 28 hours old. And even if you're not familiar with looking at these pictures, I hope you can appreciate that at least there's some similarity here. So this is a baby zebrafish. This is the eye as it's developing. So it has an eye, here's the tail, here's the head, and here's the tail. An Sf3b1 mutant zebrafish has a developing eye, has a head, has a tail. So in general, the body plan is intact. But also, even without being an expert, you can tell that these don't look identical. So clearly something is different with the Sf3b1 mutant. What you can see here, this is the forming brain and spinal cord along the back here. And this is fairly transparent, so you can really see through, showing that it's a healthy organ, an organ system. And in the Sf3b1 mutants, it's very cloudy. And what this cloudiness is denoting is actually a massive brain cell death. So we know that there's, it's the most obvious thing that's happening in these animals is that they have massive death along the brain as well as the spinal cord. So when we saw this, the first thing we wanted to know is this brain cell death apoptotic, necrotic? What kind of death is it? And can this tell us a little bit about the mechanism that underlies this phenotype? So the first thing we did was to look at active caspase 3 staining, which is going to mark the apoptotic cells. And what you can see here is that if you look at the Sf3b1 mutants, again, this is the active caspase 3 staining. It's really highlighting the brain, the eye, and the spinal cord. So although, what I didn't mention is Sf3b1 is a ubiquitous factor. Every cell in the animal has it, and it's essential in every cell in your body, my body, and all organisms. But we don't see cell death everywhere. We see cell death in the brain and the spinal cord. So this was telling us that we're seeing a specific apoptotic response. And so we wanted to know, was this apoptotic response linked to DNA damage? Because we had this, the hypothesis that splicing factors could suppress genomic instability was out there, so we wanted to know if that's what was happening. So what we did is we did a whole mount immunofluorescence for gamma H2AX, which is a mark of an active DNA damage response. And again, you can see there's really no gamma H2AX lighting up in these wild-type siblings, but you can see really massive amounts of these little spots here showing the gamma H2AX foci as they form primarily in the brain and spinal cord of these animals, suggesting they have high levels of genomic instability. So we know that the brain cell death is apoptotic. We see a DNA damage response. So is this brain cell death mediated via a DNA damage response pathway? And so to test this, we made double mutants for Sf3b1 and p53. P53 is the main mediator of a DNA damage response mediated apoptosis. So when we look, again, this is looking at Sf3b1 mutants. You see, again, this massive brain death. And if we look in the double mutants, these animals look extremely, much better, right? So the brain cell death is almost eradicated at this time point in development. Eventually, they do succumb to the loss of this essential factor. But at this time point, we know that this brain cell death is mediated by a DNA damage response. So we know that it's DNA damage response mediated, but is it R-loop mediated? Is there anything occurring in this way? All right, so to do this, we wanted to know if there were actually elevated R-loop levels in the neurons in Sf3b1 mutants. So what we did is we used an antibody that recognizes the RNA-DNA hybrid moiety of the RNA of the R-loop. This is an antibody that was developed in the 1980s, and it's called S96. It's a monoclonal antibody that's relatively selective for RNA-DNA hybrids. And what we did is we dissociated the animals, and then we stained the cells for a pan-neuronal marker, HuC-HuD, which is also called ELAV. And so we can distinguish neuronal cells from non-neuronal cells, and then we asked, so what are the R-loop levels between neuronal cells and non-neuronal cells? And are they different between mutants and wild-type animals? So what you're looking at here, this is a immunofluorescence, a single-cell immunofluorescence. This channel is looking at R-loop levels, and so in the siblings, there are R-loops that are present in these cells. Each of these little foci are the equivalent of an R-loop. And you can see in the Sf3b1 mutants, we see elevated levels of these R-loops. And then if we co-labeled for HuC-HuD, we can then distinguish what are the R-loop levels in the neurons. So while we do see kind of throughout all cells, neuronal and non-neuronal, Sf3b1 mutants have about a 1.5 to 2-fold increase in R-loop levels in those cells. We see this really, it's even greater in the neurons compared to other cells in the animal. Yes? Right, we don't know, but I'll tell you in the second half, the way different neurons respond, there's definitely different responses in different neurons. Whether the R-loop levels are different between motor neurons versus cortical neurons, I don't know yet, but I'll tell you a little bit how we got at the answer. Okay, so up to now, we have now lots of correlation. We have R-loops are elevated, DNA damage is increased, neurons are dying. But are the neurons dying? And we know the neurons are dying from an apoptotic-mediated death. But are they dying through an R-loop-mediated effect? So in order to answer this question, we utilized a transgenic system that our colleague developed. And it's based on this concept. So I mentioned earlier that RNase H enzymes are specific, they specifically destroy RNA and the RNA-DNA hybrids. So several years ago, the Crouch group figured out that you can overexpress RNase H enzymes to destroy RNA-DNA hybrids in R-loops. And this would allow you to distinguish if your phenotype that you're observing, elevated DNA damage, for example, or DNA damage breakage, if this phenotype is caused by R-loops, right? So it's one way to try and get at the underlying mechanism of your phenotypes. So you can overexpress RNase H enzymes, and this should restore, in these cases, it did restore genomic integrity when you deplete the cell of R-loops. One of the caveats with these experiments, as you can imagine, there are other processes that might be RNase H or RNA-DNA hybrid dependent, and overexpressing high levels of RNase H in cells constitutively leads to cell death on its own. So you have to be careful with this reagent. And so what our colleague did is he generated a very nice system that allowed us to transiently overexpress this enzyme during development inside of the zebrafish to determine how overexpressing RNase H would impact downstream phenotypes. If you overexpress it the whole time, the animals just die. So you definitely have to modulate the levels are really important. So what we did is we generated Sf3b1 heterozygous animals that express this construct, which allows us, this is a heat shock inducible promoter, so all we do is we heat up the developing zebrafish to 37 degrees. Normally they're grown at 28, and this will induce a heat shock response. So this promoter then turns on the expression of RNase H1, which is fused to GFP, so we can visualize the animals that have the transgene versus those that don't. And the other nice component they added, and this was based on Bob Crouch's work, is that they mutated the first they removed the first 27 amino acids, which are important for mitochondrial localization, so that this RNase H doesn't destroy mitochondrial gene replication. And it was shown in prior work that that was really important for diminishing the toxicity from overexpressing this enzyme. So when we overexpress this form of RNase H1, we then, we do this very early in development, at the onset of gastrulation. We induce expression for an hour, and then we can separate the GFP positive animals from the GFP negative animals and then assess downstream phenotypes. So the first thing we needed to know, one is to show that our R-loop staining was accurate and that the RNase H does what it's supposed to do. So what we did here was, again, we just did, this is now, we've just changed the R-loop staining represented here in pink. If you look at non-transgenic siblings, if you look at the cells in those animals, their R-loop levels are high, like I showed you in the previous slide, but if you overexpress RNase H, it greatly diminishes them. In this context, we don't get it all the way back down to wild-type levels, but it still greatly diminishes the R-loop burden in these Sf3b1 mutants when we overexpress RNase H. So we know that our reagent works as anticipated. So the next thing we wanted to know, I showed you before that whole mount in situ or whole mount immunofluorescence showing the gamma H2X levels were elevated. So we wanted to know if this was diminished, if that DNA damage response was indeed R-loop mediated. So if you overexpress RNase H and then we looked at gamma H2X, this is what it looks like in the Sf3b1 mutants that are negative for this overexpression construct. But upon RNase H overexpression, we see a really significant diminishment of the gamma H2X levels, demonstrating that the R-loops are indeed mediating the DNA damage response. And then the last thing we showed was, so we came from this to try to understand what was causing the brain cell death in the neurons of these Sf3b1 mutant zebrafish. And so we did, in this case, we wanted a more quantitative assay, so we used flow cytometry to quantify the active caspase 3 levels. And what it's shown here is that upon RNase H overexpression, we greatly diminish the percentage of apoptotic cells in the Sf3b1 mutants. And we already know that this is mostly neurons, so we know that it's diminishing the neuronal cell death. So you'd asked about specific neurons. So this was great. This told us that R-loops were doing something during early development, and that was mediating a DNA damage response. But I mentioned earlier that R-loops have been implicated in ALS. So we wanted to look at a more specific neuronal population that was more disease relevant. And so we decided to look in motor neurons, which are defective in diseases like ALS. So just to give you a quick update on how we're thinking about motor neurons and what we're thinking about in terms of R-loops with this. So as probably most of you know, I guess a mixed crowd, I don't know who knows what, but motor neurons connect the central nervous system to muscles to help innervate and move muscles forward. It's already been shown that R-loop imbalance is thought to contribute to motor neuron diseases. I mentioned earlier there are these R-loop regulators. So this is a gene called, a factor called synataxin, which is mutated in an inherited form of ALS, ALS4, specific subtype. And so it's been shown more recently that there is R-loop contribution in this disease. It's not just a suggestion correlation, but rather that it should be and somehow impacting the gene expression in ALS cells. And then more recently, there are people who have been trying to model spinal muscular atrophy, which is another ataxia disorder that in this IPS model, R-loops do accumulate. So they have R-loops accumulation, but they don't know if they cause any of the phenotypes. So we kind of wanted to throw our hat into this ring. So just before I show you our data, I need to give you a little background on understanding how we look at motor neurons in developing zebrafish. So we're looking at here, this is a schematic drawing showing a cutout of the somite of a zebrafish. This is the muscle tissue, and this is the spinal cord. So the primary motor neurons form early during development, and then the axons will begin sprouting from the spinal cord to innervate the muscles at around 17 hours post-fertilization. And so we can visualize this axon growth using a marker called SV2, which is a presynaptic vesicle marker. And so with this, we can then measure axon projections from the primary motor neurons and try to understand how Sf3b1 could impact that and how R-loops may be impacting motor neuron axonogenesis. So the first thing we wanted to know is there a defect in motor neuron axonogenesis? And the reason we picked motor neurons because of ALS, but there was also a recent paper in like 2017 showing that another splicing factor mutant had a specific defect in motor neuron axon growth. It wasn't, their motor neurons formed normally, they just didn't have, they had axon growth defects. This is why we specifically started to look at axon growth defects rather than just pan motor neuron inflammation. So what you're looking at here, this is a confocal image of a zebrafish spinal cord. This is marking that SV2 marker. This is the body of the motor neuron, and then this is the axon that's starting to sprout out. And this is all done when the fish is about 1 day old. So this is, so what my student did was to measure the axon length across the entire spinal, the trunk part of the spinal cord, and the sibling animals versus Sf3b1 mutants. You can look at the graph, but I think in this case it's relatively clear. The Sf3b1 mutants, they form motor neurons, but they really don't, the growth of the axons is greatly diminished. They have these little nubs. Sometimes they don't have any axons whatsoever. So clearly axonal growth defects are occurring. Because of this link between ALS and motor neurons, we wanted to know if R-loop, and we already know that there are R-loop perturbations in these animals to see how that was contributing. So again, we use that RNase H overexpression system that I mentioned. So we overexpress RNase H early during development, and then she measured axon growth. And what you can see here, so these are the transgenic negative fish. Again, this is what I just showed you previously. Sf3b1 mutants have greatly shortened axons. But if you look in the RNase H positive overexpressing fish, you now start to see really significant axon growth. And that's just demonstrated here with the graph. So we know that R-loops are contributing to the axonogenesis defect. But mechanistically, we're not sure how. So the first experiment we wanted to know, if you have massive cell death in the spinal cord, that's just going to make the environment kind of cruddy. Also, maybe the motor axons are just dying, so they're not forming axons because they're dying, right? So that could be, it could be really an artifact and a secondary impact, not something specific about R-loops. So in order to answer this question, we already knew that the apoptosis in Sf3b1 mutants was suppressed by p53 loss, right? So if we have a p53 Sf3b1 double mutant, we suppress the brain cell death, but does this have any impact on motor neuron axon growth? If it was secondary to the death, we would expect then this should also cause improvement in axon growth. But what we saw, hopefully you can see this, you can at least look at the graph here. So if we look at the axon growth in Sf3b1 mutants comparing P53 heterozygous animals, which have a normal apoptotic response, versus those that are mutant that have a dampened apoptosis response, we really saw no difference in axon growth. So this told us that the axon growth defect that we're observing is not a consequence of genomic instability. And we have other data that I don't have time to talk about to say that it's definitely not via ATM response or ATR, which are the main DNA damage response kinases in this cascade, but it's likely due to another impact. So kind of to go over the first half of the talk, what I've told you so far, Sf3b1 regulation of R-loops does seem to be essential for motor neuron development, specifically axonogenesis, and that aberrant R-loop metabolism is detrimental to this process, and that this is not a cell death mediated effect. So we know that some neurons, so the specificity of which neurons are impacted, some neurons are exquisitely sensitive and are dying, where other neurons are surviving, but they have developmental defects. And so even within neuronal populations at the same time in development, the way they respond to the R-loops, we don't know if it's the levels are different, that we don't know. It could be levels of R-loops are different, but for sure the response to these R-loops is very different depending on the cell context. And so it's important to try and look in those specific cell types to get at this question. Okay. I'm going to completely switch gears away from neurons. Are there questions about the... Yeah.
Have you started looking at ways to try and test this hypothesis that the reason R-loops are forming more with disrupted splicing is just that there's less protein bound to the RNA? Or do you have ideas about how to...
So that there's more. So no, we haven't tested that. What can I say? We have a few favorite hypotheses that we're testing. So it could be that there's not enough protein present. That's hard because you kind of have to look on a per RNA level. So we could do something. We could potentially do something like a clip or something to get at that question in a quantitative fashion. It could also be that the spliceosome's not getting recruited as much, and the spliceosome is full of ATPases that have helicase activity. A lot of them, like the one I'm going to tell you about in the second half of the talk, they don't really know what they're doing with the spliceosome. Maybe the spliceosome is just a recruitment in terms of its R-loop regulation. It's not the naked RNA, but it's the recruitment of these helicases that are important for unwinding them that's absent. So that's another thing we could potentially test using a similar metric. So we don't know. What we'd like to do, even from a basic standpoint, so for example, for the motor neurons, since we now know, so there's other data we didn't get, I don't have a chance to tell you, we know that the motor neurons, it's not DNA damage. So then is it because the R-loops are important for gene regulation? So it's that epigenetic modulatory component. And so to get at that question, we've now generated transgenic fish that we can sort out the motor neurons. So then we can look plus minus RNase H to see how do the R-loops impact transcription or the transcript levels, right? And then we can look at how it impacts splicing. So then we can kind of ask those questions about the reciprocal regulation also of R-loops on splicing, not just splicing on R-loops, which we don't know yet. Yep. Yep.
Is it possible to use the antibody against the R-loops to get some sort of more global picture if there's a specific site involved where it's just totally
Right, so using it as a way to like hook out the R-loops and then to genomically map them throughout the genome? Right, so we've tried to do that in cell culture systems. The signal to noise is very messy, and that we were doing with millions of cells, so we're not sure if we can do that in this context because the quantity of material that you need. So what we're trying to do very early days is to develop single molecule or single cell approaches to look at specific loci so that we can at least ask on a gene by gene basis what's happening in R-loops. And so we're working that out. A graduate student who's working that out in tissue culture cells first. If that's working, then we can apply it to the Zebrafish system where you would then only need a handful of cells to really address that question. So hopefully in a couple of years we'll get there to answer that question because it's really important to know that. So at least the global RNA-seq, where we do plus minus RNase H overexpression, it's a bulldozer, but it may give some hints of the important genes. Because it could be, in this case, it could be molecular guidance cues that are screwed up either in the neurons or the muscle cells. It could be, I have a colleague who's talked about potentially channels that may be screwed up that are important for the functionality. So, if we did at least that, we can get some hint at the genes that might be really R-loop regulated. But we don't know yet. Yeah. Any question?
Oh, yeah.
Right.
I'm curious what you think about the reason for that. Whether there's some kind of buffer against R-loop-mediated transcript.
Right, so I think there's a few points here. So yes, this is developmental. It's also not neurodegeneration because it's formation. And so we're not seeing axons grow and then get destroyed per se, although there's some concepts where you can have, it's lack of a few denervation or a few innervations that then triggers the whole death issue, so that maybe lack of innervation is the first step and then death is later. Why do people get it later in life rather than getting it now? So this is an extreme case. So I think also the R-loop perturbation is giant, which is why we can see it when we just do, IFs in the whole nucleus. Whereas if you look in a person, in the case of synataxin, which is the gene that's probably the best one linked to R-loops that is also found in that context. If you're mutant, so if you're a mutant mouse for that or a mutant fish for that, you're viable. So that's probably a more subtle perturbation because there's many helicases that are important for R-loop, for suppressing R-loop accumulation. So it might just be it takes time to accumulate, so it's slightly off, but not completely, blown up like this, which is why I think it's more subtle. That would be my best guess. And so that's why it takes time to accumulate, which would then would be more in line with maybe a DNA damage hypothesis, but our data suggests that there's definitely other things that can be involved, which might be more transcriptional or epigenetic, which could also take time. So those are my best thoughts as to why there's a big difference between these two contexts. So we have synataxin mutant fish, which we could maybe study. They're really normal, disappointingly normal. But we'll see. Are there other questions on the neuron stuff? Yeah. Yeah. Right, so we haven't, Sf3b1 mutants are homozygous lethal at like 3 days of life, so we can't study the homozygous mutants in adulthood. The heterozygous animals are fine. Right, so that's harder to do in fish. It's very easy to do it in embryos, not that easy to do in adults. But what we could, what I would say, I think if you remove Sf3b1 completely in an adult cell, the cell's just gonna be dead. Because the reason that I didn't mention this before, the reason the fish are even alive because they're lacking this essential gene is because they have maternally contributed Sf3b1 that's normal, and that eventually goes away. That takes about 3 days to go away, and we can tell this by doing, looking at protein levels and RNA levels. So I think in an adult cell, it's just gonna be dead. So I don't think, so this is a model to understand how R-loops are impacting neurogenesis, but Sf3b1 per se hasn't been implicated. Other splicing factors are implicated. Splicing changes have been implicated, but I don't know if it's going to be Sf3b1. So the one that kind of got us thinking about this is a factor called SFPQ, which is in the spliceosome, but what it does in there is kind of unclear. So I don't know. So again, those could be just milder things that allow a human to live long enough to develop these diseases. We can ask questions at the end. I just want to switch gears. So now instead of thinking about neurons, we're going to talk to you a bit about the blood system and really a completely different way that R-loops may be impacting the hematopoietic system in development. And so this one we're going to talk about a factor called DEAD-box Helicase 41. And kind of again how we got inspired to study this factor was about 4 years ago. This factor was found to be mutated in inherited forms of myelodysplastic syndrome. So these are germline mutations that people have their entire lives. They're heterozygous. These germline mutations tend to be very early. They're early truncation mutations that kind of kill the rest of the protein, but they're we think they're haploinsufficient. There's one good copy and one bad copy. There's also somatic mutations that arise in a second allele, oftentimes that hit the ATP helicase domain. These mutations are thought to be hypomorphic in function. So again, these are all thought to be things that lower the function. They're not They're not oncogenic. They're not gain-of-function mutations. These are loss-of-function mutations. And the reason that this got us really excited, because this is around the time we were really thinking about R-loops, and DEAD-box is a helicase that, let me just, sorry. So what is DEAD-box? So it has a helicase domain, it has an ATPase domain, but what is its known functions? So again, this was pulled out in this first description of its mutational status in MDS and AML. What was shown is that they did pull down through DEAD-box using nuclear fractions, and it's highly abundant. The number one thing that they pulled down were spliceosome, so it seems to be interacting with the spliceosome. What it does in the spliceosome is really unclear. The other thing that's really known about DEAD-box is that it's thought to be a nucleic acid sensor that is important for activating immune pathways upon viral infections, or also certain infections from bacterial infections as well. And more recently, and this has shown RNA-DNA hybrids that form when a retrovirus is present in a cell, DEAD-box combined to those RNA-DNA hybrids. So this is the part that got us really excited. So we're like, well, this is something that's mutated in MDS. We think our loops are going to be important in that disease, although it hasn't been that clear. And now we have a factor that's one of its putative functions, still untested. So first thing we needed to know was, does DEAD-box have a phenotype in vivo? So there's this clinical correlation that it's contributing to human hematologic malignancies, but we need a way to formally test it, and no one has done this before. So there's no in vivo models yet to study DEAD-box in hematopoiesis. So this is one of the first ones. So orient you a little bit on how we study hematopoietic stem cell development. So again, it's not the adult, it's not disease, but it's really trying to understand how this factor is important for the functionality of these stem cells that are really important for the disease. So during development, the stem cells emerge from the vasculature. So this is a picture at around, let's go through the time points first. From about 24 hours till about 48 hours, this is the time when there's a major burst in hematopoietic stem cell formation during zebrafish development. Where and how stem cells form, blood stem cells form in a developing zebrafish is the same in chicken, it's the same in mouse, it's the same in humans, so the process is the same, so we feel like this is a nice conservative system. So what we're looking at here, this is, I'm going to show you a video if it works. Yes. Okay, this is a picture of, this is the dorsal aorta, this is the dorsal aorta of a developing zebrafish, and this is the region where hematopoietic stem cells emerge. The endothelium are shown in red, the blood cells are shown in green, and so what you're going to see that the hematopoietic cells form via this process where they look like endothelial cells, change their gene expression, and become hematopoietic. So you'll see yellow cells that then change and bud out and are hematopoietic stem cells. Okay, so there should be some forming around here. Bloop, there's one. And there's another one. It just really emulates one of the real reasons I love working in zebrafish, is you can just see everything that's happening in real time. It's very powerful. Is this, oh, I think it just looped. Okay, hold on. If I stop it, it usually kills the whole presentation, so I'm going to let it finish its video. Okay, so that, just to give you some framework, that's how hematopoietic stem cell forms. We also use, oops, I'll show you this. We also use, so what this picture is, this is an in situ hybridization. So there's three different main ways that we look at HSC formation. So one is looking at the expression of key transcription factors that are important for this emergence. The one I'm going to show you the most today is RNASEH1. It's a transcription factor that's also mutated in leukemias, and it's important for this formation event. So what we started with is at around, time point, sorry. This is around, oh, there it is, around 40 hours post-fertilization. So it's kind of in that sweet spot where you're having this major burst of HSC formation occurring in zebrafish. And we're looking at here is in situ hybridization for RNASEH1. And this is what it looks like in a wild-type fish. And this is what it looks like in a mutant fish. This is for DEAD-box. So this is a surprising increase in HSC production. This is extremely rare. There are many ways that you can, there are many different mutants that you can look at for HSC formation in a fish and see less HSC formation, because kind of anything, lots of things that make the fish really sick and the vasculature sick are going to cause less HSC formation. But causing more HSC formation, there's like three three mutants and that have been studied in all of time that actually do this. So this was really surprising to us. Sf3b1 mutants, for example, make no HSCs. They can't make them at all. But that's because of the splicing defect, which I'm not going to talk about today. Okay, so they have more HSCs, which we thought was really striking. If you're not a trained professional, this is hard to see. I'm sure you're squinting. Really, is that more? Is that more? Another way to take a more quantitative approach is to use flow cytometry. And so we have several transgenic zebrafish that express fluorescent markers selectively in the hematopoietic stem and progenitor population. And so two of those transgenic lines, one is RUNX1 promoter driving expression of the M-cherry fluorochrome, and the other one is a gene called CD41, which is driving GFP, and this is highly enriched for the hematopoietic stem and progenitor population. And what we can see here is that we have a great increase in the number of HSPCs by this metric, and this is the number of cells per embryo that we can measure. So you see this huge increase. But what is the underlying mechanism? So we were already thinking about R-loops, so we were completely biased, and we decided to look to see if there was any change in the R-loop levels in DDX41 mutants. And so same experiment I showed you before, we did S96 immunofluorescence on sibling cells as well as DDX41 mutant cells, and we see a real marked increase in the amount of R-loop accumulation that's occurring in DDX41 mutants. So then, again, I've already explained the system. We use the same system. In this case, they're DDX41 mutants, and we were able to induce overexpression of the RNase H GFP as before, and then separate mutants from wild types and non-transgenics from transgenics, and then look at various assays to see how this might be impacting HSC formation. So what we did is, again, we induced heat-check overexpressed RNase H1, and what you can see here is that the number of HSCs in a DDX41 mutant embryo is around 70. When you overexpress RNase H, it's not a complete reduction back to wild-type levels, but it drops them down to about 50. So it's a significant decrease in that expansion phenotype that we observed. And this is just showing a second metric to show you that we can have two different assays. All right. So how? Why? Why are the R-loops diminishing these HSC expansions? So the first thing we did, and this is not with RNase H expression, this is just trying to understand what is the landscape of the genes that are differentially expressed between the mutants and the wild types, and there's some hint in there that can tell us what's happening. So if we look at the genes that are downregulated in DDX41 mutants and the genes that are upregulated in DDX41 mutants, we see many pathways that are interesting and important for normal development. But the factor that caught our eye the most, which I know is not the most striking one, we do see spliceosome. There are splicing changes. We don't know. They're very different. The splicing changes that happen in DDX41 mutants are very different from those in Sf3b1. But the thing that really caught our eye was this interferon alpha response, and that's because we knew that DDX41 supposedly functions as a DNA sensor. And the other reason this was important to us is that it's been shown maybe six years ago that inflammatory signaling is really important for HSC emergence. Excess inflammatory signaling, especially interferon signaling, will cause an increase in HSCs, and things that diminish inflammatory signaling will block their formation. So we knew that this was an important process for HSC emergence. So we haven't gotten our RNA-seq data back, but we did do, the poor man's RT-qPCR to tell us, are any of these inflammatory genes R-loop responsive? Can we tell what's happening? So this is just validating that what we found in our RNA-seq data, this is a list of several interferon-inducible genes. And then what we did is we looked in the plus-minus RNase H context. So if you look at the clear bars are the ones that the RNase H overexpressing, and comparing the red bars, solid red to hollow red, you can see that many of these genes really are diminished upon RNase H overexpression, suggesting that indeed the reason that you see an excess in inflammatory response is because of the excess of R-loops. So why might that be the case? So it could be direct. It could be that the R-loops are directly inside of these inflammatory genes and regulating their expression. The other thing it could be is it could be indirect. And this is was interesting to us because R-loops are implicated in autoimmune disorders, which are known to have aberrant immune responses. And so this is a simple schematic kind of outlining the pathway that we chose to interrogate. So this is the nucleic acid sensing pathway. So CGAS is an enzyme. It's cyclic GMP-AMP synthase. And what this is an enzyme that is known to respond to RNA-DNA hybrids. If they bind to it, it'll activate this enzyme. And then this enzyme produces a second messenger, which is cyclic GMP-AMP. That will then bind to and interact with an endoplasmic reticulum protein called STING. STING then triggers a signaling cascade via this kinase called TBK1 kinase, or tank binding kinase 1. And then this will lead to activation of the transcription factor NF-kappa B, which is a master regulator of several immune responses. And then this leads to activation of NF-kappa B within the nucleus, which can activate, in addition, it'll activate type 1 interferon and therefore a type 1 interferon response. So the first thing we did is we knew we had this interferon response, but do we have an NF-kappa B response? So what we did is we used a transgenic zebrafish that expresses GFP under an NF-kappa B regulatory element. So this is reading out NF-kappa B activity. And hopefully you can see this is the sibling control, and this is a mutant animal. You kind of see elevated NF-kappa B everywhere. And then if we use flow cytometry to quantify the number of NF-kappa B high expressing cells, we see this really high increase of this activity in the DDX41 mutants. So we wanted to know if this was functionally important, and so I outlined that pathway to you. So we decided to see if this NF-kappa B response was indeed a CGAS and STING mediated NF-kappa B response. Is it because of the nucleic acid sensing pathway? So we used antisense oligonucleotides called morpholinos that are commonly used in fish to knock down components of the pathway. So we knocked down CGAS and STING, and then we looked at the NF-kappa B GFP levels in the mutants and the wild types after we block CGAS or STING activity. And so we can see here, again, this is looking at NF-kappa B GFP levels. If you, in DDX, this is just showing the DDX41 mutants, and then the wild type data are shown here in the graph. What you can see is there's a severe, a very significant decrease in the NF-kappa B GFP activity in the CGAS morpholino animals as well as the STING knockdown animals, suggesting that the inflammatory response, the NF-kappa B inflammatory response, is indeed mediated via the nucleic acid sensing pathway. So that's true. That's great. It's kind of already known that that's important for that inflammatory pathway, but isn't going to be important for the HSCs. We know that the R-loops are affecting HSCs. Is this inflammatory pathway also impacting HSC expansion? So we did the same experiment, knocking down CGAS and STING, but now we looked at how this impacted HSC levels. So for these experiments, again, we saw, if you look here, this is the level of HSCs in the DDX41 mutant animals, and then when we compared this to CGAS morpholino injected animals or STING morpholino animals, we see really a significant decrease in this HSC expansion. There's some variability in which pathway member we knock down, but we definitely see a great decrease in this. So then the last experiment we did was to try and do a genetic interaction study. So we know that R-loops are doing something. We know that CGAS and STING are doing something. But are they working through the same pathway? Is this actually R-loops going through CGAS, STING, and then leading to this HSC phenotype at the end? So this experiment's a little complicated. Hopefully I can take you through it. So what we did in these experiments is we used our RNase H overexpression fish. So we inject the morpholino. In this case, we used the STING morpholino. So we knocked down STING, and then we also overexpressed RNase H. If they're working through the same pathway, then if our hypothesis is correct, STING would be downstream, R-loops would be upstream. If you get rid of STING, doesn't matter what you do to the R-loops, the STING phenotype is going to be epistatic to the R-loop phenotype. Right. If they're acting through separate pathways, we might expect to see an additive effect where you knock down R-loops and you knock down STING, you'll have a greater reduction of HSC levels. So what we saw, the graph's probably the easiest thing to follow. So these are just looking at DDX41 mutants. If you look at the HSC levels in the mutants, we see some diminution in the R-loop overexpression system alone. But when we have STING morpholino, whether they have R-loop depletion or not, it's the same level of knockdown. So we're definitely seeing that there's a huge, the STING is epistatic and downstream, that's our interpretation, of R-loops, so that they probably are working through the same thing. So I think this is what I just said, but I'll just clean it up a little bit so you can follow. When DDX41 levels are down, R-loop levels are elevated. This R-loop accumulation leads to an inflammatory response via the CGAS-STING pathway, and this then leads to HSPC expansion. What we don't know is this something that happens in humans? And so, like I said, there's no models where anyone else has modeled this in the mouse system. It's really poorly studied in the human system to understand if there are indeed elevated R-loops in the human system, is this increased inflammation intact? So hopefully what I've just told you, two very different stories. R-loops are connected. We know from the neurological side, some neurons are very sensitive to R-loops, R-loop-mediated DNA damage and neurodegeneration. Others are impacted by differentiation. In the case of our hematologic system, we've shown that inflammation is a major trigger. But what we haven't really looked at is the flip-flops on either side. So one, do Sf3b1 and Ddx41-regulated R-loops in the same way? Do they regulate the same R-loops? I like to think yes, but I have no evidence for that. It's just a hope. They could be different subsets of R-loops that are in different genes. How does R-loop imbalance regulate the expression of sulfate and differentiation factors? And so that's kind of the stuff we talked about in the first half. Is it really direct? In the case of motor neurons, since it's not DNA damage, could it be direct regulation of those genes? Also, in the motor neuron context, we haven't yet looked. Maybe it's inflammatory. We don't know. There is elevated inflammation in Sf3b1 mutants as well. Could that be, and it's known that neurons are hypersensitive to inflammation, and so it could be that this is also occurring in this context. We just haven't explored it yet. Is there a role for the DNA damage response in the hematologic phenotypes? And so we do have some data that at least ATR activation is involved with the HSC expansion phenotype. So there could be some crosstalk amongst these many different flavors of how R-loops are impacting different tissues. But without looking at each of the aspects, it's hard to know. All right, and then thank all the people in the lab who did the work. Sarah Nick is here. She's actually left the lab and works at Selectus up in the city, so she's moved on to different science. Josh Weinreb is a graduate student who's finishing up now, who did all the work on DDX41. Nora Ghazali is a student. She's a shared student with Rob Singer's lab who's trying to figure out how to actually look at R-loops in single cells and also to take on the neuron project so that we can kind of take it to the next level. We did the first part of the talk was done in collaboration with Rod Stewart's lab at University of Utah, and Varun Gupta and Charles Creary's lab has helped us with some of the bioinformatic analysis so far. So thank you. Any other? You asked a lot of neuron questions. Yeah,
so the signaling cascade, the CGAS-STING signaling cascade, everything that's happening at the protein level, right? It's not gene regulation until you get to NF-kappa.
That's how it's the signaling pathway as it's activated by exogenous. So what I would say is from exogenous RNA-DNA hybrids, that is thought to be the case. It's exogenous things come in, bind to CGAS, and then activate the cascade, and nothing changes transcriptionally until NF-kappa B. In our context, because we have endogenous, it's really endogenous R-loops or endogenous RNA-DNA hybrids that are mediating the response. We don't know if it's going to be two hits, so to speak, so that there may be R-loops in inflammatory genes that is direct, and there's this cascade that gets triggered in the cytoplasm because these things leak out in some unknown way, but they can leak out and then trigger this response. We don't know.
I was wondering if you could look in like RNA-seq from MDS patients and see whether you see those genes, that signature of that signaling.
Right. So it's so what I would say is there is in MDS patients there are signatures of an interferon response, right? So that exists. There's signals of NF-kappa B activity, whether it's via CGAS or not. So STING and TBK1 are slightly elevated at the RNA level in MDS patients, but I don't know if that means the activity is higher. You can look by phosphoflow, so we can look at, that hasn't been done, but you can look at phosphoflow for some of these specific downstream pathways to see if the pathway is activated in that context, but things to do. Christian, maybe? Mm-hmm.
Are R-loops, are there a consistent number of R-loops across all tissues, or are these tissues enriched for R-loops relative to others, or are there enriched tissues?
Great question. I wish we knew. So we were. So Nora's original project was to try to make a way to look at R-loops in real time in fish because the antibody or Whole immunofluorescence does not work. It doesn't tell us anything. We can't see. You really have to like zoom in at 100x in order to see those nuclei to know that you're looking at a specific signal. So we don't know. We'd love to answer that question. What I can tell you is in the blood system where we can fractionate out cells so we can get stem cells and then get differentiated cells. Differentiated cells in the blood system have like 0 R-loops. They have none. I mean, it's striking. They really have nothing, but they're really enriched in the progenitors. So we think, again, hand waving, the chromatin state is very different. So in differentiated cells, the chromatin is very shut off. There's only a few loci that are really active for that specific differentiated gene, cell type, whereas in the progenitors and stem cells, it's more open. And during development, the neurons are all going to be kind of in open state because it's very developmental. So great question. Hope we can answer it in a few years, but yeah, we're still working on that. Thanks. Thank you.